So welcome back again, whether you took a full break or whether you just clicked through to continue on with the next this recorded session. Let's take a look at the concept check, answer these quick questions, and then we can move on to talk about mitosis. So a chicken, like all birds, has 78 chromosomes in its somatic cells. How many of these chromosomes came from each parent? How many chromosomes would be in the chicken's gametes? How many chromosomes would there be in the somatic cells of an offspring of the chicken? Well, remember that somatic cells are diploid. They have two complete sets of chromosomes. So to find out how many chromosomes came from each parent, all we need to do is divide 78 by 2, and that will give us the half number of chromosomes, or the number of chromosomes in one complete set. And of course, 78 divided by 2 is 39. So half the chromosomes, or 39 of the chromosomes in each somatic cell, came from one parent. 39 of the chromosomes came from the other parent. How many chromosomes would be in the chicken's gametes? Well, that's also going to be 39, because we know the gametes have one complete set of chromosomes. They're haploid. And in this case, the haploid number would be half of 78, that's 39 again. Have many chromosomes would there be in the somatic cells of an offspring of the chicken? Well, that offspring should have the same number of chromosomes as its parental chicken parent. That would be 78, because that is the chromosomal number of all birds, including chickens. They have 78 chromosomes. So there we have it. Chickens have 78 chromosomes, and therefore the offspring of this chicken would have 78 chromosomes. So hopefully you got those questions right. Very important concept here, the understanding the nature, the difference between somatic cells and gametes, something that we're going to hit home again pretty hard in the next chapter. But we need to talk about mitosis here. So when we start talking about mitosis, the actual process of cell division is not the equivalent of mitosis. So one of the important things to remember here is that mitosis and cell division are not equivalent. So mitosis is division of the nucleus. And it results in two nuclei. And actually, I'm going to erase that and say two identical nuclei. And if you want to know what this is step 2. Hopefully you remember that. Step 2 of the 3 steps. Cytokinesis is the actual division of the cell. And is separate from mitosis. And it's actually step 3 of the 3 steps. So obviously what's missing is step one, and we know that step one has to precede the division process because it's the duplication or the copying of the DNA, and we'll see where that takes place in a moment. But this is something that really gets freshman biology students into trouble, trying to make this equivalence between mitosis and cell division. And even a lot of biology professors and textbooks don't do a really good job of separating mitosis and cytokinesis from each other, and separating mitosis from the stage of the cell cycle called the mitotic stage, which covers both mitosis and cytokinesis. So what I think I'll do, put these together. And this is the reason why there's this little bit of confusion, because we divide the cell cycle, as you'll see in a moment when I show you the diagram, into two basic stages. The stage during which cell division occurs, which is referred to as the mitotic stage, and then everything else, which is referred to as interphase. And so because we put mitosis and cytokinesis together within the mitotic phase, there is a tendency, I'm not sure what to call it, but just say a tendency to think of mitosis and cell division as the same thing. The mitotic phase and cell division are the same thing, but mitosis, no. Mitosis is simply division of the nucleus. The rest of the cell divides during cytokinesis. Why this confusion? Because it just so happens that cytokinesis, as division of the cell proper, occurs in parallel with the last step of mitosis. And because they seem to occur together in parallel, it's very easy to say, oh, well, cytokinesis is part of mitosis. It's not, and I'll keep coming back and saying this over and over again, because it's really important to make this connection and separation between the two. But it's understandable why people get confused. And hopefully that's not going to be you. And we'll try and make sure that it's not you. So let's take a look at the cell cycle itself. So this is what the cell cycle looks like. Here's the mitotic phase, which you can see is composed of mitosis and cytokinesis. And then you have interphase, which sometimes is referred to as the resting phase. And when I learnt mitosis way back in the dark ages, as I like to say, when dirt was young, we learnt that interphase was truly a resting phase, because back then, very little was known about what actually was going on during that part of the cell cycle when the cell was not obviously dividing. And eventually it was recognized that there was a very specific point in the interphase where you could actually measure an increase in the amount of DNA. And so clearly, as the DNA is increasing, so if you've got a point in time or a period within the interphase where the amount of DNA is clearly doubling, then that's obviously the point at which the chromosomes are being copied. So at this point, interphase is no longer a single period of time, it's broken up into 3 periods of time. And those 3 periods of time became known as G1, S, and G2. And G1 stands for growth. Sorry, not growth. I like to call it growth. I shouldn't be quite so. G1 is gap 1, and G2 is gap 2. And the reason why they're called gap 1 and gap 2, because they are obvious gaps in activity. There's no question, but there's activity going on when the cell is actively dividing during the mitotic phase. And now with the ability to be able to measure the increase in DNA during what is now called the S phase of interphase, you have 2 periods of activity. One where the cell is obviously dividing and one where the cell is obviously copying the chromosomes. But then we have these 2 gaps in between the mitotic phase and the S phase. So they became known as gap 1 and gap 2. Gap in activity, I guess. But the reality is these are active growth phases. But the growth is geared towards different things. So in the active growth phase G1, here the cell is recovering from cell division. It's increasing the amount of protein, the amount of membranes. The mitochondria would be replicating here in plant cells. The chloroplast would also be replicating in this period. Basically, this is the cell starting to increase in size again and starting to increase its internal contents after getting through cell division, because it's got to replace all of the material that it's divided up, or that was divided up amongst the 2 daughter cells. Now the cell's got to make up for that missing amount of material. So that's taking place during G1. That continues on during S. Don't think that there's no growth going on during S. There is. Growth is a continuous process all the way through interphase. It's just that S is a specific point within interphase where we know for certain the chromosomes are being copied. And this is all going on while the chromosomes are in their chromatin form. So you can't actually see it happening if you look in the microscope, because remember, whatever the DNA is in its chromatin form, it's invisible. So during the S phase, the only way that you have of knowing that the DNA is being, or the chromosomes are being copied, is by being able to measure the amount of DNA in there. And there are ways that we can do this. One way is using fluorescent dyes that bind to the DNA. There are other ways of doing it as well. Some involve using radioactive isotopes. Others involve physically extracting the DNA and literally measuring the amount of DNA that's in there using chemical methods. But you can clearly indicate or detect the increase in the amount of DNA during this period of time. It's what is unique about this period of time. Don't feel like there's no growth going on. There's still growth going on. It's just being that S is going on against the background of the rest of the growth that's going on that sets this time period aside. Then when you get to G2, there's more growth going on, but at this point, the cells are essentially committed to dividing. In fact, if the cell begins duplicating the chromosomes, there is an indication of commitment to division at this point. And so from this point onwards, as the chromosomes are starting to be duplicated and you go through S and continue on to G2, basically what's going on here is a preparation for the actual division. So when you get to the end of the G2 phase, there is a commitment to division. And so long as the cell is given permission to divide, and you'll understand why I'm saying that when we talk about the regulation of cell division. As long as the cell continues to get permission to divide, at the end of G2, it has everything in place to support the division process, to undergo mitosis, which remember is division of the nucleus, and then to undergo division of the cell during cytokinesis. So that at the end of the mitotic phase, you have two daughter cells that then enter G1 and begin the process of the cell cycle all over again. So this is basically how we understand cells to progress, as long as they're constantly and actively growing and dividing. Now, the vast majority of the cells in our body are not actively growing and dividing. That doesn't mean that they can't grow and divide, although some of them are probably have signaling blocks that prevent them from dividing, and we'll talk more about that when we talk about the regulation of cell division. But the vast majority of our cells do have the ability to re-enter the cell cycle and grow and divide if it's necessary for them to do so. But again, the vast majority of our cells are not actively growing and dividing. They just have the capacity to begin growing and dividing again, should it be necessary. Some cells, not so much, nerve cells particularly, don't. And there's a story behind that as well. But this is basically what happens if cells are actively growing and dividing. They participate in this cycle. So you can see that the cell division part, the mitotic part, is actually a small part of the cell cycle. So the mitotic phase is only about 10% the cycle. Cells in interphase, that's 90% of the cell cycle. And in fact, the vast majority of cells are not even in the interphase portion of the cycle. They're outside of the cell cycle, as you'll see when we talk about, again, when we talk about the regulation of the cell cycle. So this is the cell cycle as we understand it. We do need to layer on a few more details, and we'll do that as we talk a little bit more about the actual process of mitosis, and as we talk about the regulation of the cell cycle. We'll layer on a little bit more of detail about the cell cycle. But let's now focus on the mitotic phase by focusing specifically on mitosis first, and then shifting our focus to cytokinesis. Remembering again, the cytokinesis is not physically part of the mitotic or mitosis, but because the cytokinesis occurs in parallel with the last part of mitosis, it's not uncommon to see the cytoplasm dividing as the cell is completing that last stage of mitosis. So initially, when you look at cells under a microscope, the vast majority of cells that you're going to observe are going to be in interphase. If it occupies 90% of the cell cycle, even in those areas where we know that cells are actively growing and dividing, then simple statistics tells us that the vast majority of cells in that collection of cells must be in interphase. And you can confirm that because you won't see the chromosomes. They're going to be in their chromatin form during interphase. So you're simply not going to see them. We know that the DNA is being duplicated during that time, specifically during S, but you're not going to see the process taking place because it's all done while the DNA is in its chromatin form. But once the cells enter the first phase of mitosis, referred to as prophase, you start to see the chromosomes appearing. So what I want to do is give you a simple mnemonic that will help you remember the 5 phases that we recognize in mitosis. So just trying to think, yeah, I think this might be the best place to put it. So here's a simple way of remembering them. PPMAT. And so what does PPMAT stand for? So the first is prophase, prometaphase, metaphase, anaphase, and telophase. And again, for those that might be going, so why don't we have a C for cytokinesis? Remember, cytokinesis is not part of mitosis. It's a separate step. So some of you may have learnt mitosis before and learnt PMAT, prophase, metaphase, anaphase, telophase. That's because there has been some disagreement amongst cell biologists as to whether prometaphase is a legitimate stage or not. Most freshman biology textbooks include prometaphase in a discussion of mitosis. Our book certainly does, and most other freshman biology textbooks also include prometaphase as one of the 5 stages of mitosis. So we will include prometaphase. But as you'll see, there is some disconnect here. There's some inconsistency in the usage of these names, and that will become clear when we talk about chapter 13. Because we're going to revisit some of these names in chapter 13. You also may find in some of the resource videos that I've got posted on the resource pages, and in fact I know this is the case, that there is no discussion of prometaphase, that everything that the book has in prophase and prometaphase is under the heading of prophase. So that's something that you need to pay attention to as you're doing some outside reading and investigation, and if you're using the resources, which hopefully you are using, that you may not find a reference to prometaphase. But because the textbook does include prometaphase, we will include prometaphase. I just want you to be aware of the fact that there is no consistent usage of prometaphase at this point. Some people use it, some people don't. Even within the textbook, there is a little bit of a disconnect between chapter 12 and chapter 13, but more about that when we get to chapter 13. So prometaphase, prophase, prometaphase, metaphase, anaphase, telophase. Each of these phases is defined by what goes on with the nucleus. Why? Because mitosis is division of the nucleus. And it is easy to identify what's going on in the nucleus by looking at what's going on with the chromosomes. So a lot of what is used as key identifiers for each of these stages is with reference to what is going on with the nucleus or the chromosomes in the nucleus, or not in the nucleus as the case may be. So if we go back to the previous slide, prophase is identifiable because the chromatin form of the DNA undergoes that coiling and supercoiling and condenses into the chromosomes. And remember, these are duplicated chromosomes. So the more condensed the chromosome, excuse me, the more condensed the chromosomes become, the easier it is to see that they are composed of two sister chromatids, that each chromosome structure consists of two sister chromatids. At the same time, a structure built out of microtubules called the spindle starts to form. And it forms from features that are commonly referred to as MTOCs, microtubule organizing centers, but can also be referred to as centrosomes. Actually, what I'll do, I probably should do that. Let's take this out here. So it's the centrosomes in animal cells. And they contain the centrioles, which we talked about back in chapter 6. So MTOCs is a generalized term that applies to both animal cells and plant cells. But because animal cells have centrioles, we sometimes use the term centrosomes to refer to these features from which the spindle grows. At the same time, within the nucleus, we have these structures called the nucleolus, plural nucleoli. Those actually start to break down as prophase begins as well. When we get to prometaphase, and remember in some of the references that you might refer to, these features of prometaphase are actually included in prophase, but our book separates them out into prometaphase. We see that the nuclear membrane breaks down. And there's a simple reason why the nuclear membrane has to break down. Because some of the microtubules of the spindle are going to attach to the chromosomes. But in order to be able to attach to the chromosomes, they have to be able to penetrate the nucleus, because the chromosomes are inside the nucleus, the spindle is forming in the cytoplasm. So the nuclear envelope needs to break down in order for the microtubules to attach to the chromosomes, and they don't attach just anywhere. They attach to structures called the kinetochores. I'll put that underneath. I may run out of room there. Maybe room a little bit there. But the kinetochores are structures that are associated with the centromere region of the chromosomes, and there are two kinetochores, one associated with each of the two sister chromatids. When we look at the diagram of prometaphase, I'll point this out to you. Metaphase is real easy to recognize because the chromosomes line up across the center of the spindle on a feature called the metaphase plate. It's not an actual physical feature, it's a virtual line that bisects the spindle, and all the chromosomes line up on the metaphase plate, and they're moved there by the actions of the microtubules that are connected to the kinetochores. In anaphase, again, there's a very simple way of recognizing anaphase. The two sister chromatids separate. The reason why they separate is because the centromere structure So up until this point, the centromere complex, which is a complex of proteins, has been holding the two chromatids together. But in anaphase, the centromere complex breaks and allows the two sister chromatids to separate from each other, and they migrate to opposite ends of the spindle following the microtubules. So the microtubules provide a framework for the sister chromatids, now chromosomes, to actually migrate along as they move to the away from each other to opposite ends of the spindle. Then in telophase, it's basically a reversal of everything that went on in prophase and prometaphase. So in prometaphase, the nuclear envelope broke down. So in telophase, it starts to form up again. In prophase, the chromosomes started to condense and become visible. Well, in telophase, they decondense and return back to their chromatin form again and become invisible. During prophase and prometaphase, the spindle formed up. In telophase, the spindle breaks down and disappears. So everything that went on in prophase and prometaphase is reversed in telophase. At the same time, and very typically at the same time, in parallel, cytokinesis begins, which is the actual division of the cytoplasm. Remember, that is separate from the mitotic phase, or from mitosis. Let's not say the mitotic phase, but separate from mitosis. But it does occur typically in parallel or in conjunction with telophase, which is the reason why it's very easy to make the assumption that cytokinesis is part of mitosis. Just make sure that you don't fall into that trap. So here are the diagrams. And again, the artist has tried to make it a little bit clearer for us in terms of what is going on here. So while they've drawn the DNA as thread-like, remember this is strictly invisible while ever the cell is in interphase. And of course, G2 is that last part of interphase. So this is what the cell would be like as it's getting ready to begin mitosis. So we tell that mitosis is beginning because the chromosomes start to become visible inside the nucleus. And you can see that the chromosomes are just starting to become visible. And the spindle's starting to form up. And here, the spindle microtubules are stained yellow, so it's real easy to see them. You can see the centrioles within the centromere, the centrosome. Too many centro terms, sometimes you get a little bit confused, even I do. So the centrioles in the centrosomes are visible here, again stained yellow because they're made out of microtubules. They start to move away from each other, and you can see them moving away from each other. They're moving in opposite directions. And as they move, the spindle starts to grow. The microtubules start to grow out of the centrosomes, and the spindle starts to form a cage-like structure that eventually surrounds the whole of the nucleus. Then as the nuclear envelope starts to break down, some of the microtubules then attach to the kinetochore structures. So microtubules from this end of the spindle attach to the sister chromatid on this side of the pair via its kinetochore. Microtubules from this end attach to the other sister chromatid. So each chromatid is attached to one end of the spindle. One member of each pair is attached to one side. The other member is attached to the other side of the spindle. And then as the microtubules continue to grow, they jostle the chromosomes around and move them towards the center of the spindle. And you can see now why it's an advantage to have the chromosomes in the condensed form, because it just makes it so much easier to move them around attached to the microtubules. Eventually in metaphase, the chromosomes have all reached the center of the spindle. They're all lined up on this virtual line called the metaphase plate. This, by the way, also represents the point in the cell where cytokinesis will begin. This is where the cell will actually divide. along this line. So it's not just an arbitrary line where the chromosomes line up. This also represents the plane where the cell is actually going to divide when cytokinesis begins. So then we move on to anaphase, where the sister chromatids separate from each other, and each starts moving, guided by the microtubule or microtubules attached to its kinetochore. It's now moving away from its sister chromatid, but now we have to refer to them as chromosomes. So rather than being sister chromatids, they're now daughter chromosomes, separating from each other and recognizing or identifying and acknowledging that they have separated from each other and they are identical to each other. So they are daughters of the same chromosome. They are now separating from each other. Notice that not all of the microtubules are necessarily attached to chromosomes. We'll talk a little bit more about those in a couple of minutes. So after the chromosomes reach the ends of the spindle, they start to cluster together and start to decondense. At the same time, a new nuclear envelope starts to form around them. A nucleolus starts to appear, or several nucleoli, depending on the type of the species and types of cells, start to appear. And the spindle starts to disappear and break down. So all those things that we saw going on in prophase and prometaphase, they're getting reversed. And at the same time, cytokinesis begins. Now, this is cytokinesis in an animal cell, and that's relatively easy to understand because it simply involves a cell pinching in half. It's a little bit more complicated than that, but the evidence is that the cell pinches in half, and you can see it beginning here as the cell begins to pinch in half. So those are the 5 stages of mitosis: prophase, prometaphase, metaphase, anaphase, and telophase. So there are your 5 phases. And remember, in parallel with the 5th phase, telophase, you also see the beginning of the division of the cytoplasm, otherwise known as cytokinesis. So don't confuse cytokinesis with mitosis. Make sure you keep them separate. So what is the role of the spindle? Well, we've seen that the role of the spindle is to guide the movement of the chromosomes, capture the chromosomes, move them to the center of the spindle, ultimately in metaphase, and then to guide the sister chromatids after they separate from each other, what are now daughter chromosomes, as they migrate to opposite ends of the spindle. No mystery there. And so here you can see the microtubules and you can see the kinetochore complex that they've attached to. It's a little bit difficult to see the separation of the 2 sister chromatids, but that's the line of separation between the 2 of them right there. But as you can see in this picture, there's only a limited number of chromosomes, but there's a whole bunch of microtubules. And as we've seen before, it's really unusual to imagine that a cell would make something if it didn't serve a purpose. So the question then becomes, what is the purpose of these microtubules? And the answer is they rub against each other, or they push against each other to elongate the cell along the long axis of the spindle. So these microtubules are stretching the cell this way. And the artist has actually represented that back here in these diagrams. Notice we've gone from a circular cell to something that's more ovoid in shape. That's the effect of these microtubules. They are stretching out the cell, and you might ask, why do that? Simply to create more room, to create a greater separation between the 2 daughter nuclei, so that when cytokinesis really does begin, there's room for the division to take place, and to further guarantee that each of the 2 daughter cells gets one of the 2 nuclei. It's just a way of guaranteeing that there's room for division to take place. And these overlapping microtubules, the so-called non-kinetochore microtubules, are what stretch the cell out. But again, that shouldn't come as a surprise, because these are just microtubules doing what we've seen microtubules do all along: change the shape of the cell or provide a pathway for things to move along. And here we can see these microtubules doing both. They guide the movement of the chromosomes, and they stretch the cell out to make it a bit easier for cytokinesis to divide the cell into 2 halves. Now, it used to be thought that the microtubules actually pulled the chromosomes apart. You can still see that reference. Even the textbook kind of suggests that. The truth is that nothing further could be the case. The microtubules are not pulling the chromosomes apart, even though it may look like that. What they're actually doing is allowing the chromosomes to walk along the microtubules because of the kinetochore. There are motor proteins in the kinetochore. And motor proteins, you may recall, are what guide things along microtubules. They interact with microtubules and guide things along microtubules. And so the motor proteins inside the kinetochore are simply walking the chromosomes along the microtubule. At the same time, there's an enzyme in the kinetochore that disassembles the microtubule after the chromosome has walked past, because now its job is done. There's no point in preserving it, and so it gets disassembled. And this diagram, and you can read about this diagram in the textbook, it's taken straight out of the textbook, simply illustrates the research that went on to demonstrate this. So these microtubules were stained with stains, the yellow stain. Then in this region, a laser beam was used to destroy the stain. And as you can see, rather than the region where the stain had been destroyed by the laser beam moving, it's the chromosomes themselves that move. If this region moved, then we would know that the chromosomes are pulling. But because it stays stationary and only the chromosomes move, then it's the chromosomes that are literally walking along the microtubules. They're not being pulled along the microtubules. And we now know why. We know the function of the kinetochore in that. Then the other thing that we need to bear in mind is that plant and animal cells cannot do cytokinesis in the same way. And you might ask, why? Well, the answer is simply because plant cells have a cell wall that must remain intact for this plant cell to function properly. Animal cells don't. So animal cells simply pinch in half, and that is the function of a ring of actin proteins, otherwise known as microfilaments, that simply pinch down in the center of the cell where the metaphase plate is located, that virtual line, and they pinch down in that region and simply pinch the cell into two daughter cells. It's pretty straightforward. As far as plant cells are concerned, it can never be that simple, because you need to have an intact cell wall at all times surrounding the cell. So the way cytokinesis takes place in plant cells is that the developing cell wall that's going to complete the surrounding of the two daughter cells is what actually divides them in half. So it starts out as a series of vesicles that line up along what was the metaphase plate. They are producing cellulose fibers that are going to build the cell wall. These vesicles get bigger and bigger as they produce more cellulose and start to expand towards the ends of the cell and out to the side, and forming this structure called a cell plate. And that cell plate expands and expands until it contacts the existing cell wall all the way around. And as it contacts and fuses with the existing cell wall, it completes the division of the parental cell into two daughter cells. And at no time did the parental cell or the two daughter cells lack a complete cell wall surrounding them. And so the formation of the cell plate and its growth into the new cell wall carries out cytokinesis. And you can see here in this electron microscope image, the vesicles lining up in the center in between the two nuclei that have formed up. This is going to grow into the cell plate, which will eventually divide the plant cell into two new daughter cells. So here are some pictures. The previous pictures, when we talked about the phases of mitosis, were of an animal cell. So here are the same phases in a plant cell. So prophase, prometaphase, metaphase, anaphase, telophase, and you can see the cell plate forming up, which tells you that cytokinesis is also, again, occurring in parallel with telophase. And if we were doing wet labs, we would actually use onion root tip cells, some commercially prepared slides of onion root tip cells, to look at these phases of mitosis, because we can guarantee to find cells dividing in onion root tips and root tips in general, because the roots of plants along with the shoots of plants grow at the tips. So the tip of the root is where you will find dividing cells that are causing the plant root to grow. And the tip of the shoot, the top of the shoot, is where you'll find cells that are dividing to allow the plant to grow from there. Plants don't grow from the ground upwards. They grow from the tip of the shoot upwards. Nor do the roots grow from the bottom of the shoot downwards. They grow from the tip of the root outwards. And so the tip of a root makes a good place to look for dividing cells. And in fact, it's a really good place to look for growing cells because we can guarantee to find them. Something that we can't do if we were looking for dividing cells in animals. So this slide simply summarizes everything that we've learnt about the cell cycle, the fact that it is broken up into two major phases, the mitotic phase, which is the cell division stage, and interphase, which is the remainder of the cell cycle. So remember, the mitotic phase is about 10%, interphase is about 90%. Interphase is then broken up into three distinct steps, G1, S, and G2. Remember, S is distinguished by the fact that this is where the chromosomes are being duplicated. The amount of DNA is doubling in here. G1 is where the cell is recovering from division. Mitochondria are duplicating, chloroplasts in plant cells are duplicating, more endoplasmic reticulum, more ribosomes, lots of chemical activity going on, and that continues all the way through interphase. G2 is where we see a lot of growth directed specifically towards cell division. We see the beginning of that in S. So again, remember that growth does not stop just because the DNA is being duplicated. The duplication of DNA, duplication of the chromosomes, is just something that we were able to differentiate very early on because of developing ways of measuring the amount of DNA in cells, and so we could separate out this time block from everything else because we could physically detect the increase in the amount of DNA. Then when we look at mitosis, and remember that is division of the nucleus, strictly just division of the nucleus, we have prophase, prometaphase, metaphase, anaphase, and telophase. Cytokinesis then represents division of the cell proper or division of the cytoplasm. So that's mitosis and cell division. So when we look at bacterial cell division, here the situation is really easy to understand. It's called binary fission. Binary means two. Fission means to split. So this literally means to split in two. So that's what binary fission means. And this is the only diagram that you need to reference. And these two slides are the only two slides that you need to reference in terms of bacterial cell division. So as I mentioned way back at the beginning of the presentation, bacterial DNA is in the form of a small circular DNA molecule that's anchored to the inside of the cell membrane, the plasma membrane. There is a physical location on that circular DNA molecule called the origin of replication. That's where where duplication of the DNA begins. And it moves or it expands in both directions away from the origin as the DNA is duplicated. Eventually, you have two copies of the DNA molecule with two origins. And we think that the origin of replication represents an anchoring point to the plasma membrane. That's been proposed. People have gone backwards and forwards about whether that is really the case or not. But the general consensus is still, I understand from my reading of the literature, that the origin of replication represents the anchoring point. So as the cell grows, the membrane has to expand to accommodate the volume. We talked a little bit about that back in chapter 6 when we talked about organelles. We talked about surface area to volume ratios, and the size of the membrane has to expand in order to accommodate the increase in volume. So as the cell gets bigger, there's going to be a bigger membrane, and that serves to separate the two DNA molecules. So once the cell gets to a predetermined size and the two DNA molecules are completely separated from each other, the cell simply splits into two. That's the binary fission process. This is how bacterial cells divide. So if you want to tie these to the 3 steps, this is step 1. This would be step 2, and this would be step 3 of those 3 fundamental steps of cell division. And remember, bacteria invented cell division. So not surprisingly, we see the 3 steps played out. In fact, I think I'm going to change this just a little bit. Oops. And say that this is step 2. Because it's in between there that we get the separation. So that's all you need to know about bacterial cell division. So now we've talked about general eukaryotic cell division. Mitotic cell division is how it is usually referred to, and bacterial cell division. So we'll take another brief break here with, again, some concept check questions. I will stop the recording, so if you want to step away for a little while, feel free to do so. If you just want to follow on, of course, click to jump to the next and final part of this presentation. So here are our concept check questions. How does cytokinesis in animal cells differ from plant cells, and why? And during the stages of the cell cycle, does a chromosome, or when during which stages of, I should read what I've got written here. During which stages of the cell cycle does a chromosome consist of 2 identical sister chromatids? I'll see you on the other side of the break.